progress.
Okay, good morning. We will start the A2 class, and I will give an introduction to the A2 class. So, A2 class, we are going to start unit 4. So, unit 4, we have two topics, topic 5 and topic 6. So, first part is topic 5, and we're going to learn 3 lessons. Lesson number 1 is photosynthesis and ecology and climate change. Topic 6, we're going to learn microbiology, immunity, and forensic. So then these lessons going as 5A, 5B, 5C, 6A, 6B and 6C. So these are the 6 lessons we have learned under unit 5. So unit 4. So the assessment is, the paper is 1 hour 45 minutes, and the total marks, raw marks 90, and the UMS mark 120. The rest of the lessons I will also add here, unit 5 and 6. So unit 5, again 1 hour 45 minutes and a 90 mark and 120. Unit 4, unit 5, 1 hour 45 minutes, again 90 and the UMS 120. Unit 6, 1 hour 20 minutes, 50 marks, UMS 60. So total mark is 300. From this total 300, to get an A, you need to get 240, and to get an A star, you need to get 270. Right, so this is the overall lesson plan in unit 4, and we have a unit 4. The evaluation say 90 marks, raw marks, 120 UMS, 90 raw marks, 120 UMS, and 50 and 60. So 300 is the total, and to get a total A, you need AS and A2, that is 480. So this is the total marks you should get for AS and A2. Then you get an A. And exceptionally, if you want to get A star, you need to get 270 in A2. Right, that's a whole lesson plan and the marking system of unit A2 biology, right? So what we trying to do in our class work, we're going to finish the syllabus in January. So that is 2027. And after that we're going to do exams and practice, paper practice until end of May 2027. January, March, April. This most probably February. And we have 4 months for paper practice and exams. Even we can do 1 more class in June because Unit 4 is Unit 4 and 5 it's already finished now, and Unit 6 is still there to do this year. So that's the plan we're going to do in the class. And we have a first lesson photosynthesis, and what we do, I will give you a printed paper, a book actually, with notes and the exam practice question. We do regularly exam practice question in the class, and then only the paper practice we start in February. Full papers we start in February. So if you have any question, you can ask. What we did, the lesson plan for A2 and what we're going to do in A2. So again, summary is, we have a Unit 4, we have 3 lessons, and we did this: photosynthesis, ecology and climate change, and topic 6, microbiology, immunity and forensic. The evaluation is Unit 4, 1 hour 45 minutes is the paper, and this time the biggest problem students face the time management, because the paper has 40 pages, Unit 4 paper. So comparing Unit 4, Unit 4, 5, 6, which is the hardest paper is Unit 4. Even the lessons are simple: photosynthesis, ecology and climate change. But the hardest paper is Unit 4. You will see why. Unit 5 basically about the human body, that is the muscles, coordination, nervous system, biotechnology, and that Unit 5 paper and 6 paper is quite easy. Right. So the total marks you can get again 300, the raw UMS mark, and 240 is A, 270 is A star. So we are going to do the first lesson, photosynthesis.
Sir, for seeds, that's like the subtopic, like for A,
These are like ABCs.
Yes, sir.
That within topics, Unit 4 has 2 topics,
Yeah.
5 and 6.
Yes.
So here I delivered Unit 5, topic 5, 5A photosynthesis, 5B ecology, 5C climate change. Then again 6A, 6B, 6C.
So, sir, for 6C, it's forensic, it's only like a small part, right?
This one is not a big lesson. This one is small lesson, but microbiology and immunity, it has a lot of things to study.
So, sir, like, it doesn't exactly teach everything, like it only teaches the beginning of forensic science, right? That's what
Forensic science, we only focus on the most important lesson. Okay, I just give a small introduction. What is forensic science? Forensic science is dealing with the crime scene. Crime scene, simply, people kill people. So this is happening everywhere in the world. Some countries less, some countries more. But when a person has been killed, the biggest problem is, who is the person killed this man or woman? So we have to find the person involved in crime scene. So despite of all this technology developed, people still try to escape and difficult to find. Then this forensic in EDexcel only dealing with mainly time of death. So how can we find time of death? This is the biggest lesson we're going to learn. Because if you know the time of death, then it gives a huge evidence to find the person involved in the crime scene. So that's a small thing, but the problem is, so you have also set for this AS unit 1, 2, 3 now. One thing is, when you see the paper, unit 1, 2, or especially 1, 2, or 3, you see it's quite new, like new lessons, new formats. Whatever you have done past papers, you always see in the exam new formats and new questions and new patterns. And the same thing happened in unit 4. Then I asked student what lessons came. Forensic, no sir, this time no question from forensic. Then microbiology, a little bit, immunity little bit, then this a little. So this time unit 4 paper, they add only a little thing, not big questions, and more application. So most of them say unit 4 is hard compared to unit 5 is easy, because it's mostly about the human body and they can answer. But unit 5 is dealing with climate change, because climate change is a big concept, and also ecology is a big concept, and these things, when it come diseases and microorganisms, they are also evolving, like new things are coming. So keep in mind, unit 4 you have to focus a lot than other units. So as I told you, I will also give for each topic a printed note, and also questions that we need to do this question in the classroom, because not like AS, the unit 4 questions are quite challenging question. You can't write using the only theory knowledge. You know the theory, but still you can't write the answers. But some questions yes, some questions no. So that's important to practice more questions in the class from each lesson. And from these 3 lessons, photosynthesis, ecology and climate change, easiest one is the photosynthesis, because it's very straightforward. Concepts are very solid, straightforward, and only you need to understand and remember the things. Right? So let's start. It's a 5A. The number is 5A. So you must be wondering that what should we learn from photosynthesis. So it's always, for us, it's a small lesson. Just a very small lesson, and many times we are trying to write all the reaction, carbon dioxide, water, and it produces glucose and oxygen. So balancing equation is 6, 6 and 6. So we all know this from a primary level. This is all about photosynthesis. then when you think, okay, A2 level, what we are going to learn about photosynthesis? It's a very simple thing. But when it comes to photosynthesis, this is the most complicated things in the world, and many rich people like Bill Gates, they're trying to understand this process because all the people fighting for energy. The energy is the most valuable thing at the moment in the world. So when it comes to energy, if we can know all the things happening and we try to use this artificially, we can produce energy. Because if you see the glucose is energy molecule, but you will see somewhere in the middle, we can also produce hydrogen. So hydrogen is again a very good fuel source. So people trying to understand this process, still they did not discover everything. So unable to discover everything, because if we know everything, then we can artificially produce the hydrogen as a fuel source, and then we can produce enough energy. Or even we can produce glucose as a food, and glucose we can chemically combine to make starch. Then food crisis is also solved. So the biggest problem, energy and food crisis in the next coming up years. So evidence, yesterday I saw United Nations, their report say 3 countries are in danger in food. They are running out of food. What are 3 countries? Yes, Afghanistan, Ethiopia, Sri Lanka. I was shocked because we have a lot of food outside. Australia, Delgediac, at least we can eat and live. But I don't know what kind of criteria they use to evaluate this. Right, now, when it comes to photosynthesis, we started with how we can define it. We can define it is a process in which sunlight energy, the pure energy source is sunlight energy, is used to produce the energy molecule glucose or starch. That's a very simple definition. So who can do this? And then we have to define the group of organisms that can do photosynthesis. The main organisms are plants. The rest, very little bit from prototists, and very little bit coming from bacteria. So main source is the plants. So then we consider plants as autotrophs. So autotrophs are organisms that they can do photosynthesis. Then what are the other groups? Other groups are heterotrophs. Heterotrophs are human and animals. They eat the producers for their energy source. So autotrophs use sunlight energy and carbon dioxide as a carbon source to make the energy, and this energy usually trapped between carbon and hydrogen bond. So when we use this glucose or starch, then glucose starch energy is again converted into chemical energy, ATP, adenosine triphosphate, in the cells. This is a universal energy currency. That means all the organisms use ATP as the energy source in the cells. So this is the introduction, and we are going more advanced concept of photosynthesis in the next lessons. So can you please write the topic today's 5-day photosynthesis? Then we make a simple definition of photosynthesis. So photosynthesis is a process. Photosynthesis is a process in which the energy of sunlight, energy of sunlight converts to chemical energy such as glucose or starch. In the presence of carbon dioxide and water. So if we think about this chemical reaction, 6 carbon dioxide, 6 water, and 1 glucose molecule and 6 oxygen. So chemical energy produced is 2880 kilojoule. So that's as per this reaction, the energy level is 2880 kilojoule. Right? So can you write this chemical reaction?
So that's endothermic reaction.
Yes, energy absorbed. 12 was plus side and minus plus side.
So that's 602, right?
602. But you can write, continue writing about these few things. Then you write next thing, autotrophs. Are the organisms who can do photosynthesis. They are plants, prototists, and some bacteria. Next one, heterotrophs. Heterotrophs are the organisms who eat plants or primary consumers for their energy source. And the last point about ATP, the main energy source in the cell is adenosine triphosphate. Adenosine triphosphate. That is, within bracket you can write ATP. We'll also look a little bit why ATP is the universal energy source in all organisms, ranging from bacteria to large animals. So, a little bit about the ATP molecules. So ATP molecule, it's very similar to a nucleotide. It has a ribose sugar and 3-phosphate group, 1, 2 and 3. And here nucleotide adenosine. So it's just very similar to a nucleotide, except it has an additional 2-phosphate group. Otherwise, this is adenosine nucleotide. And other difference is in DNA, you know, it's a deoxyribose, but this is a ribose. Now, how this molecule is important? This molecule can hydrolyze. Right? So hydrolyzed to produce adenosine diphosphate. So this bond hydrolyzed and one phosphate is released. So it becomes adenosine diphosphate. So this is very easy reaction in the cells because it need to break the bond we need energy. Right? So to make the bond we also need energy. So breaking and making we need energy. So still the energy source we can minimize by adding enzymes. That is the enzyme used here, and using ATP ATPase enzyme can break it and release this phosphate, and it can produce around 34 kilojoule per mole. So it can produce 34 kilojoule per mole of ATP. Then to make ATP, we can again go this direction. It is a condensation reaction, and the enzyme is ATP synthase. So the two reactions, hydrolysis and condensation. So this is a very established strong reaction in the cells. So that's why all the organisms they use ATP as an energy source, and it can get a very high amount of energy. Not only that, it can recycle. So it can recycle and it's available, it's not running out. So cells can recycle it, and this is we're going to learn in unit 5, the respiration, how cell produce ATP using glucose. So glucose is a sole energy for all of us from the plant, and glucose break down to produce ATP. You will learn the glucose molecule completely break down into carbon dioxide and water. All the carbon and hydrogen bonds break, and that energy release, this energy is used to make this reaction. So then they produce ATP in the cell. So can you write this, draw this diagram? diagram, ADP is universal. ADP is universal energy source in the cells due to following facts. Due to following facts. Number 1, rapid turnover. Rapid turnover. Rapid turnover. In front of that you can write it can convert to ADP. It can convert to ADP. To release the energy. To release the energy. And resynthesize to produce ATP. The second point, the reaction is rapid. The reaction is rapid and less overheating of the cell. The reaction is rapid and less overheating of the cell. Number 3, it's a small and soluble molecule. It's a small and soluble molecule. Right, that's the end of these basic lessons. So we have learned what is photosynthesis, autotrophs, heterotrophs, and the value of ATP molecule. With this knowledge, we are going to the next level. Where does this process take place in the cell? So we all know this process takes place in the cell in chloroplast. So we need to know when we learn about photosynthesis, the structure of the chloroplast. Where this happens and how this happens is the next process. So now,
recording in progress.
Right. Next thing is the structure of chloroplast. This is very important thing. You can write the next topic. Because all of the reactions of photosynthesis take place inside the chloroplast. So that's why it's very important to know the structure. So when you want to know the structure about this, it's like an oval shape and it has 2 membranes. That's a cell membrane and that's the outer membrane. Inner membrane, and in between the space is called intermembrane space. Everything inside is a jelly-like structure and it is called a stroma. Right. Next thing is the most important thing. So inside the stroma we can find the disc. So this disc appears like this and they make a column stacked together to make a columns like this. The one disk is called tidal coil. The collection of the coil into a column is called grana or granum. That is something to do with singular and plural. The granum is singular, grana is a plural. And what else we can find? Elsewhere, we can see this kind of a structures. And here, I just show only 3 columns like this. And they are interconnected. So the interconnection is called lamellae. And we can also find another few things. That is a 70S ribosome and circular DNA. And sometimes we can also find a starch grain. And the size of the chloroplast is roughly around maximum maybe 7 micrometer. And length and width is around 2 micrometer, 2 to 3 micrometer. So this is also important because you will see in the practice question, you have to identify some of the parts in the exam. Right? Identify this, what's the role of this, like that. And not only that, when we're going to learn the reactions, we should know what is stroma and tidal coil. Which reaction take place in stroma and which reaction take place in tidal coil. That's also important, right? And also, I don't have in the notes, probably I will give the books. And online, when you join, I send the PDF file, just you can print it and keep with you, because at least next week we starting questions. Because you need to know first the theory, then we can start the question. So we should also identify this from the electron micrograph. So that's 2 things, right? So I think no need to write every function of these things, what is this, but you just draw this, because when you're going to learn the reactions, we specify in which place this reaction take place, and the most important thing is the tidal coil, because most of the important reactions happen inside this tidal coil, or the membrane of the tidal coil. Right? So take your time and complete this diagram. Right, shall we write the most important thing that we need to remember about a chloroplast, the structure? The main component of the chloroplast is thylakoid. Thylakoids, which are organized into columns known as grana. You can write to the same place. The thylakoid membrane is packed with pigments which can absorb sunlight. So we will see details later about the pigments. Now pigments can absorb sunlight because we know that energy can transfer in terms of waves, like light waves, sound waves and electromagnetic radiations. They all transfer energy. So this energy can be harvested or absorbed by different methods. Pigments are one of the key molecules that they can absorb pigments. We can find two types of pigment. One is called main pigment, just key pigment. You will understand later why this chlorophyll A. So chlorophyll A is a molecule and this is the main pigment. The other pigments are accessory pigment. Accessory pigments are chlorophyll B, carotenoid, pyrophytin and xanthophyll. So we can find many pigments because when we look at the plants, sometimes you see different color of the leaves, like a yellow color, slightly white color like this. So when they have these pigments, we can also see different color, but chlorophyll A mainly gives us a green color. So chlorophyll A we can see as a green color because you will see next lesson light, the visible light consists of different colors. These pigments can absorb different colors and emit what is they can't absorb. So that means chlorophyll can absorb almost all colors like red, green, yellow, orange, like all these, and emit green color. So basically the energy in these wave forms absorbed by the pigments then only the pigments start chemical reactions to produce energy. So the chlorophyll A, and these are the main pigments and accessory pigments. So I think you just write this as a table. So just briefly write what their role, and you will better understand later. So involving main reaction. So they absorb and harvest the light. And the chlorophyll A also absorbs the light, and then it is going to the main reaction. Which is note here, harvest the light, and this is involving the main reaction. Chlorophyll A involving the main reaction. So this chlorophyll B, carotenoid, and pyrovitin xanthophyll, they can harvest the light. In other words, if you want to understand what they are actually doing, this is something similar to this. If you take a magnifier and you hold this magnifier in bright sunlight, and you can harvest the light into one spot, and it's a very bright spot. So this can even burn the papers. Like if you hold for several minutes, even you can burn the papers. That means you focus light energy to one particular point, and the energy is very high here. So these pigments also organize to harvest the light. So when they harvest the light, there should be some kind of damage can happen because it's too much energy. You will see they harvest the light, and they bring to the chlorophyll A. So then the chlorophyll A will not damage. So there is a participant question. They ask, question is, what is the role of different pigments in chloroplast or thylakoid? Describe the role of different pigments in the thylakoid membrane. So we should know, we should able to answer this question, and then we write a small heading here, the role, the main role of the pigments, the main role of the pigments. The first one you will write, that is a preferred marking scheme point. Right? So the first, I will also give 2 additional points, but first one is the main marking scheme point. So it is 2 marks or 3 marks maximum question. You will write answer like this. Different pigments can absorb different wavelength of the light. This resulted absorption of higher energy and increased rate of photosynthesis. Second point is important to know, but we will not use as main important marking scheme point. And you can write second role of the pigments. Excessive light is absorbed by accessory pigments which prevent damage of chlorophyll A. Right, next thing we should also know the visible light wavelength and which pigments are good in absorbing the visible light. So this lesson now goes as absorption spectrum. So absorption spectrum. Absorption spectrum. So here the x-axis we place the wavelength of the visible light. So wavelength of the visible light ranging from 400 nanometer to 700 nanometer. And y-axis we place here arbitrary unit or percentage of light absorbed. For example, if it is 100%, so any pigment which can absorb 100% of light, it goes here. So that's the absorption. We take only the 3 pigments. The main pigment is chlorophyll A. So chlorophyll A, they can absorb light. The colors are here is a blue, violet, right? So violet, indigo, blue. then this is a green and yellow, orange and red. So otherwise, this is maybe blue color area, and that's a green color, and that's a red area. So chlorophyll A, they can absorb here, and nothing much here, and again this is chlorophyll A. Chlorophyll B can also absorb light here. It goes here, and so that's chlorophyll B. And carotenoid, it just absorb and nothing much here. Just want to have a rough idea, never asked to draw this kind of diagram. So the main point is the blue color and red color is the main, and blue color is the first one, and the red color is the second one, and green color absorption is very less because it's emit the green color. So this is called absorption spectrum. So can you sketch this diagram?
So there's a small peak in orange, right?
Which one?
In the graph, there's a small peak in orange, right?
This is the highest one represents B, and here highest one is A, and this one is just.
You said that the peaks, the small peaks are at orange, right? In the area.
Yeah, this range.
Yeah.
Yeah, basically we call actually the blue color and red color. Like in general, we don't specify exactly this violet is this nanometer and that's excel like this. We are not going that. We just going the highest light can be absorbed at blue color and red color. For example, practically you want to have a greenhouse and just you want to grow plants. Then you go for like a blue color light, so it has more energy. Okay, we just write the definition of absorption spectrum. When you write here, or maybe what is this graph? Absorption spectrum represent absorption of the light, at different wavelength by different pigments. We can also understand if they absorb more light here, then more light is available for photosynthesis. Then this area, actually no more light absorbed, and there is no more photosynthesis with this wavelength. And again we can find some amount of the light energies here. That means they also used to make this light energy for photosynthesis. Then we can also draw another graph similar to this called action spectrum. So action spectrum represent the overall rate of photosynthesis with different wavelength of pigment. So it's like this. So this is called action spectrum, and we write here rate of photosynthesis. Yeah, that's also important, the action spectrum, so you can also copy this down. shall we write the action spectrum definition on this? So action spectrum represent the rate of photosynthesis at each wavelength So one more thing to finish. After we learning one important thing, then only we going to learn the reaction of photosynthesis step by step. So there is a small thing to learn that is called photosystems. Systems. PS, abbreviated by PS. Okay, now what is photosystems? They are collection of pigments, different pigments, to harvest the light. So where we can find them, I just draw a thylakoid. So this is thylakoid membrane, and this thylakoid membrane is just a normal cell membrane, and in this cell membrane we can find different photosystems.
Recording in progress.
Okay, let's continue with the last part, right? So if you remember cell membrane, we can find the channel proteins and carrier proteins. Similarly, we can also find in this lipid bilayer this kind of a photosystem scattered everywhere. And there is another group of proteins you will also see in this membrane, and these two components, that is the photosystems and proteins, which are called electron carrier proteins, they are organized everywhere in this kind of thylakoid membranes. So let's look a little bit detail of this photosystem. Now I am using a different diagram because the photosystems are 3D shaped spherical-like complexes, sphere-like complexes. But to understand the purpose, I try to draw it like this. So it has a component like this, and another component like this, and another component like this. So basically, how it's located, it's very difficult to draw 3D structure. So it's like this, in 3D form, like this. So I hope you can get some idea about these 3D shapes. So something like this, and 3D shapes. The photosystems are like this. They have different components, and the middle, they have a one component. So I am showing one component and the middle component, right? So here we have pigments and also pigments here. So these pigments are accessory pigments. So then we can call these complexes light harvesting complex. So I mentioned you, when there is excessive light, the excessive light can transfer here and transfer here, transfer like this. They can move to different pigments, so it will not damage. Because if there is one pigment and you take all the light, you can understand it can damage because of too much light. Then here we have a main pigment, that is chlorophyll A. Now you can see very carefully light is dissipated and moved to this chlorophyll. Same thing can happen here. Okay, now if the light is absorbed by the molecules, what will happen to the electrons? the energy is used by the atoms, and the final energy is going to the atoms. And when it is loaded with energy, who would like to leave first? Electrons. You know that when you get energy, electrons jump to the next shell like that, and the energy of the sunlight excited the electrons. Then electrons get this energy, electrons leave. So I can write this very basic equation like this. Chlorophyll A, after getting this energy, it becomes chlorophyll A plus and electrons. Because it lost electron, the molecule get positive. Now here, the place is called reaction center. And electrons now move into the reaction center, and somebody should accept electrons. So reaction center has primary electron acceptor. So how photosystem work now? The light, we call it, let's say call it photon. The light energy comes as a photon, and that energy is absorbed by the accessory pigment in the light harvesting complex, and then it moves the chlorophyll is the main pigment. Chlorophyll A excited, and the electrons released, and the electron is going to the reaction center. Reaction center has the primary electron acceptor that accepts the electrons. So one more thing now left. When the molecule becomes like this, they are not stable, because the stability came when they have full electrons. Once electron left, these molecules are not stable. So we'll see how it goes later. So that's the photosystems, and we have two types of photosystems. One is called photosystem 1, and the other one is called PS2. Why we have a two different system? Because PS1 absorb best at 700 nanometer, and this absorb light at best 680 nanometer. Because they have the same complex and same appearance, but the only thing is what they have the pigments, how much light they can absorb depend on the PS1, PS2 complex. Otherwise, the basic structure is same. So that's the last thing we should know to understand reactions about photosystems. So can you copy this diagram?
chlorophyll beam. Side columns, the dots are chlorophyll beam. No,
The light harvesting components. These two. So they are arranged like this. Light harvest. And then you bring to the chlorophyll A in the middle, this one, and the reaction center. So shall we write a small note for this, and then with this we are ending the main the basic things which we need to know, the reactions of photosynthesis. So we write like this: photosystems are located in thylakoid membrane. They have a light harvesting complex which absorb light and pass the energy to chlorophyll A. Pass the energy to chlorophyll A. I think you can understand why chlorophyll A is main pigment, because even though they pass the light, the electrons with energy is released by chlorophyll A. So that means just to understand, like if you remember about the nuclear reactions, the uranium atoms are hit by the neutrons. So neutron is actually activated. Same way, everything starts with the chlorophyll A by releasing the electrons with energy to chlorophyll A. Then next thing you write, then chlorophyll A molecules are excited. Chlorophyll A molecules are excited and release electrons to the reaction center which has the primary electron acceptor. which has the primary electron acceptor. Yes, so again, when we're going to learn the reactions, the sunlight energy going to this place, so the energy is now with electrons. So now electrons leave with the energy, then next all the reactions start because of these electrons. So that's what I want to show you, that how the uranium, the atoms activated by neutrons, then all the energy start to release. Right? So far we learned the basic component that we need to understand the rate of reaction, the reactions, and now we are coming to the main part in the next lesson. So it's just like all the little component we learn, we're going to focus to the advanced concept of the reactions in the next lesson. So what I do, I will give you an overview of photosynthesis, and then I take one by one each thing, then we learn in details. So this is A2 with advanced. And you will see the contents are more than A's, that you need to remember more than A's. Just go to the summary of photosynthesis. Now, photosynthesis, the reaction, we can divide into two. The first reaction is called light dependent reaction. That means the first stage, definitely they need the light. Then the second reaction is light independent reaction. So light independent reaction, it doesn't matter whether light is there or not, or light or dark, it just, this reaction can happen with light or without light. So first light dependent reaction happen, then this start. So this reaction is also called Calvin cycle. Now, light-dependent reaction, we can again divide into two. One is called cyclic, or just like first one, non-cyclic. Non-cyclic. Cyclic. Okay, this word I will introduce you is called phosphorylation. The other one is cyclic phosphorylation. So what's the meaning of phosphorylation? If there's any molecule and if we can add a phosphate group, so this reaction is called phosphorylation. Adding phosphate group, so example ADP plus phosphate group, ATP is phosphorylation. So this is the phosphorylation. So good example is ADP plus a phosphate group to make ATP. Right. So this is the overall reaction, and then if you want to still see what is the end of the reaction, so end of the reaction is you would like to see glucose, because you know the reaction end is glucose and oxygen. Right? Oxygen is a byproduct, so we not focus on oxygen, and it produces oxygen. Now the next point, why light dependent reaction is important, because light dependent reaction produces energy. This energy is now going to this Calvin cycle or light dependent reaction to produce glucose. Then they can fix carbon, hydrogen, oxygen, because when they have energy. So that's how the whole photosynthesis work. First, light dependent reaction take place. Why? Because it needs to produce energy. Light energy is used to produce ATP. Then ATP is required to produce glucose in this light independent reaction. Then they can combine carbon, hydrogen and oxygen, because carbon is coming from carbon dioxide, oxygen is coming from water and carbon dioxide, and hydrogen is coming from water. So these atoms fix together to make glucose if we have energy. So that's the overall reaction and summary of photosynthesis. So what we do in the next lessons, we're going to learn this in detail, especially this one. So this is the most important reactions, non-cyclic phosphorylation. This will also touch a little bit, not much. As we learn this one, this is just a small thing, like it's a small version of this. And then next we're going to learn this reaction. Then it's a N, we will see glucose. So most complicated one is this. Still, we learn it, right? So can you draw this overview of the summary of this? Then you know where we are when you go into the lessons. So what I want to tell you again, we have this topic 5 for these lessons, all the lessons, and limit for topic 5, the notes and the exam practice questions. So why exam practice questions are very important? Because when you look at these questions, there are more application type of questions. So if we learn pure theory, it's useless. So we need to practice questions. And another thing, this also includes the practicals. Unit 6, we have again 9 core practicals. So we have to also do the questions of the practicals and also statistics. So we are covering statistics in ecology lessons, next lesson of photosynthesis, because it's involved the statistical test. There are 3-4 tests you have to learn: t-test and chi-square test and Spearman rank correlation test, like this. And we should also practice exam paper questions in unit 6. Otherwise you cannot get enough knowledge to handle the questions if you do it at the end. So the flow is, for example, the core practical, there's a core practical for this lesson, photosynthesis. There's a small one, I'm not going to much focus. Chromatography is there, but chromatography, you know all these things. The how to measure the rate of photosynthesis is a core practical. So we learn core practical and immediately we do the past paper question based on this practical. So this also includes here, right? So that's very important. So please, the online when you join, so I think you can print it and make the book of this, and okay, please write the next lesson, light dependent reaction. Next lesson is light dependent reaction. So I will also show here the complete pathway of electron transfer, and then we take one by one and we're going to learn what is happening at each level. So this is the thylakoid membrane. The thylakoid membrane is a lipid bilayer. So if I show it here, 1 phospholipid and 2 phospholipid, the whole membrane consists of phospholipid and it makes a bilayer. So the starting point is the PS2, and there is electron carrier proteins, and again there is transport and electron carrier proteins, and again electron carrier proteins. Then we can find the PS1 complex. PS1 complex, again electron carrier proteins, and electron carrier protein and enzyme complex. So these are the main components, and you will see the electrons excited from the light energy transfer to these complexes. Then we are going to learn what is happening at the end of the electrons, what will happen in the middle. So it's very important, this electron released with energy, sunlight energy. So electrons moving with energy, this energy can be used at different points to make another type of molecules. So we don't have much time, but we will continue next week, but you can copy this down. So I just give A, B, C, D and E. A, B, C, D, E. Electron carrier proteins, transport, enzymes. Yes. So we don't want to know the names of A, B, C like this. Just we understand the electron carrier proteins. I mean they can carry electrons or transfer electrons, or they can act as a transporters. For example, B could be transported because it's expanding from here to here, and some may be enzymes. And PS2 and PS1 is the photosystems, and this represents the thyroid membrane. Okay, we'll continue next week the lesson by dependent reaction, and see you again next week.
Go.